Hello everyone, today we will discuss neurolation, how it works and how it develops different organs during embryogenesis. But before we proceed, we will just try to familiarize first these different terminologies for us to understand new terms as we go on to our discussion. So what actually is neurolation? Neurolation is one of the events of embryogenesis, by which the neural tube develops and furthered with the primordium of the central nervous system, including the brain and the spinal cord. The process of neurulation is divided into two, which are the formation of the neural tube and the formation of the neural crest. So the first thing that we will discuss is the formation of the neural tube, which is divided into two phases, the primary and the secondary neurulation. When we talk about primary neurulation, it is when the cells surrounding the neural plate proliferate, invaginate, and pinch off from the surface to form a hollow tube. And we talk about the secondary neurulation on the other hand, it starts after the neural folding and formed in the tail bud, which wherein the spinal cord initially forms as a solid mass of epithelial cells, and the central lumen develops secondarily by cavitation. So, primary neurulation starts within the third week of development, and it is formed in the anterior portion of the epiblast or the surface of the ectoderm. So, as we can see in this illustration, there is a blue color structure, and this is what we call to as ectoderm, the surface of the ectoderm. And the hole in the middle is the embryo, or what we call the primitive node, and the structure above it, line structure above it, is what we call the primitive string. So one of the morphological sign of this primary neurulation is the formation of the neural plate, wherein there is a dorsal thickening in the anterior ectoderm concomitant with the regression of this primitive streak above it. So as we can see in the other illustration, there is a what we call notochord, which is located below the ectoderm. And this notochord induces the formation of the central nervous system by signaling the ectoderm germ layer above it to form the thick or flat neural plate. So this neural plate undergoes in shaping, so it converts it into a more illuminated keyhole-shaped structure with broad anterior and narrow posterior regions, which leads to the formation of the two lateral elevations, the what we call neural folds and the neural groove. So what is neural folds? Neural folds are the two left and right which rounded the neural groove. So the neural groove, as we can see, is the hollow-like structure in the center. So after these neural folds continue to elevate and oppose in the midline, it eventually fused to create the neural tube, which becomes covered with a surface ectoderm that will develop into the future epidermis. So in the primary neurulation, there is a bending of the neural plate and the opposition of these neural folds. And it has the principal sites. Take note of these. The first is the median hinge point, overlying the notochord, and the paired dorsolateral hinge points, or the DLHP, at the points of attachment on the of the surface ectoderm to the outside of each neural fold. And after these neural folds, here comes the next phase, or what we call the secondary neurulation. So in secondary neurulation, it is when the posterior portion of the neural tube is developed, and it is formed by the neural fold, which will fuses together by the cell surface glycoconjugates. The anterior and the posterior most part of the neural tube communicates with the amniotic cavity through these two openings. So what are the two openings? mainly the cranial or the anterior neural pore and caudal or the posterior neural pore. And when the anterior neural pore is closed at 24th day and posterior neural pore closed at 28th day, it is a time when neural tube is complete. So as you can see in the illustration, the central nervous system is represented by this time by a closed scapular structure with a narrow posterior portion. Wherein the anlage of the spinal cord and the much broader cephalic portion, the primordium of the encephalon, or what we call the brain. So during neurulation, the neuroepithelium is entirely proliferative, which means that cells do not begin to exit the cell cycle and start neuronal differentiation until the neural tube closure is complete. And for the secondary neurulation, we'll just remember also the fusion of this neural tube, which begins in the cervical region and proceeds anteriorly and posteriorly from there. This fusion is mediated by the cell surface glycoconjugates. As is a result of these processes, the neural tube is formed and separated from the overlying ectodermal sheath. We all know that neurulation is a critical event in embryogenesis, and that's what we mentioned in the primary neurulation during the formation of the neural tube. There are different morphogenic movements like elevation, bending, adhesion, or even fusion of cells in the surface of ectoderm. Therefore, apoptosis during neurulation is very prone, which leads to the cell death, disturbs anterior neurulation, reduces spinal neural tube defects, probably by preventing this dorsal midline remodeling, and the failure of closure results in the neural tube defects, or the NTDs, like exencephaly, or in the cranial region, and spina bifida, resulting from the failure of this spinal closure. So for us to understand more and easier, let's try to figure out this big concept. So first is the neural plate, or the dorsal thickening of the surface of the ectoderm, which give rise to the elevation, the neural folds and the neural groove. And after this neural folds and neural groove fuses in the midline, it will give rise to the neural tube. And this neural tube will give rise to the epidermis after it will be separated from the ectoderm. And this epidermis will further be the primordium of the brain and the spinal cord.